Well, with an introduction like that, I'm kind of stuck under pressure. I should actually call this the electric human, because I want to talk about electricity, and not just water, but electricity. Let me tell you just how we got started. It started with two famous scientists, one of whose picture you've just seen. But the first was Albert St. Georgi. And St. Georgi is considered to be maybe the father of modern biochemistry. And a brilliant fellow with wonderful quotes. He's highly quotable. For example, life is water dancing to the tune of solids. And now he knew that water was central to all of biology. And then came Gilbert Ling, who's now in his 90s. This picture was taken long ago, and he's written 5 books since then. And there were several major contributions. One of his contributions was that the water inside the cell was not like this water here. It's actually water that is structured in some way, organized. And the idea was that next to protein surfaces, which are charged and have hydrophilic regions, the water would build up into some sort of ordered structure, and that the inside of your cells and my cells were filled with water. And most scientists today don't really think of water as being important. They think of water as being, well, as I said, just like that. However, we all know that we're 2/3 water, approximately. And if you translate that 2/3 into the number of molecules, because the water molecule is so small, it turns into more than 99% of our molecules are water molecules. So they've got to be important for something. So I actually took off on this. It wasn't so long ago, about 10 years ago, starting from Gilbert Ling. And the idea that the water inside the cell was critical to any kind of cell function. And I wrote the book, and the book was originally supposed to be to translate Gilbert Ling's ideas into plain English and to try to expose them to the community. But I soon realized as I started writing that there was really more to it than what Gilbert had thought. But still, the idea of structuring of some sort of water next to surfaces was really important. Well, by some serendipity, we came upon an idea to test the idea of water structuring and how far the structure might extend from the surface. And so the idea at first was a simple one. We took a gel, in this case it was a polyvinyl alcohol gel, and we put it into a chamber, and we poured some water in, water plus we put particles, these tiny spheres, microspheres in, although later we used different solutes and got a similar result. And the idea was that Gilbert Ling brought up was that this kind of ordered or structured water would expel solutes, just like ice. And therefore, if you want to find out how extensive this region is, you can just see how far the region exists without solutes or without particles. So we did this experiment, and we found, this is the first time pretty much that we did it, is that these particles were expelled from the region near the interface. They kept moving and moving and moving, and then they'd stop at about 50 or 60 micrometers from the surface. They would undergo thermal motion, moving around, bouncing, but they would never enter back into the region, the clear region. They were excluded, and therefore we call this the exclusion zone, or EZ for short. Another example of it is shown here. And in this case, we take a polymer, naphthalene, and it comes in sheets, so we just cut the sheet and place the sheet down in the bottom of the chamber and add the water and the microspheres and see what happens. And this is what happens. The exclusion zone builds up, and it builds, and this video is cut prematurely. It'll extend out to something like 300, 400, 500 micrometers. That's practically infinite by molecular standards, huge numbers of water molecules in between. So by now, this finding has been repeated by and confirmed by many people. In fact, it was found, it was reported in the Journal of Physiology in 1970, and it was a bit embarrassing to find out after the fact that someone else had done it and got pretty much the same results. So we know that these exclusion zones exist, and the question is what's really going on? So the questions that I want to answer are, first, is this exclusion zone, this phenomenon general, or does it just apply to some polymers and a couple of gels? Does it really arise from the ordering of water? Because if it did, it would mean a huge number of molecules ordered, and this is against conventional thinking. And since creating order requires energy, where does the energy come from to create this if it really is ordering? And finally, what I want to spend most of the time on is, can the ordered zone help explain everyday phenomena? And by phenomena, I mean, for example, why do clouds exist as they do, and how come when you sprain your ankle, it swells very quickly? So the first question is the one of generality, and we've done a lot of experiments, and I don't want to bore you with all of them, but we've found that many substances show this exclusion zone, as you can see here, including monolayers. By monolayers, what I mean is single molecular layers. So you have a single molecular layer, and this exclusion zone builds, which means you need basically only a template. You don't need something that has thickness to it. And among the solutes that are excluded from this exclusion zone, it's not just the large particles that I've shown you, but solutes ranging down in size to approximately 100 molecular weight. And one example of that is shown in the next slide, where we take pH-sensitive dye. And now these dyes are mixtures of molecules whose size is on the order of 100 or so molecular weight. And it's cool because they show you local pH by changing color. And so this is an example of that. At the bottom is a piece of Naphion, similar to what I just showed you. And all the pretty colors that you see are indicating different pHs. The main point of this slide is that the region right next to the Naphion here is clear, free of dye. So it means the dyes are excluded. And as I said, the dyes are approximately 100 molecular weight. What's even more interesting than that is the color distribution. And that orange color that you see just beyond the exclusion zone means a pH of 3 or less. So a lot of protons are sitting right next to that exclusion zone, and fewer and fewer. And we'll come back to that because that's really important. So in terms of generality, many hydrophilic surfaces generate exclusion zones, and many solutes are excluded from it. Okay, so question 2 is this zone really physically different from bulk water? I've alluded to the possibility that it might be different, but I haven't shown you any evidence. Well, I'm not going to show you a lot of evidence because it will take a lot of time, but I'm just going to summarize most of the evidence and show you some of it. And a lot of the evidence is published, and the stuff the rest that hasn't been published will be in my new book, which should be out in 2 or 3 months. So the first is that these easy water molecules are more constrained than bulk water molecules. They're also more stable, we found from infrared radiation coming from it. We found that the exclusion zone has a negative charge, and I'll show you those results. We'll come back to it since this is obviously central to the interests of everybody sitting here. We found also that this zone absorbs light in the ultraviolet at 270 nanometers. That number is important, and I'll come back to that one as well. We found that it's more viscous than bulk water. We found that the molecules in that zone are actually aligned in some way. And we found that the molecular structure is somehow different from bulk water. And finally, this is not our results, but the results in 2 Russian groups who looked at the optical properties of the exclusion zone. And they found that the refractive index is about 10% higher than that of bulk water. So there are a lot of things that are different in this zone from bulk water, so it's clearly a distinct zone. Now, going back to the negative charge that I mentioned, the way we did this experiment is we set up a chamber, and where it says inside, that's actually the inside of a gel. So you have a gel sitting there, which in this case is a polyacrylic acid gel. And outside refers to outside the gel, where there's water. So we took 2 electrodes. These are microelectrodes that taper down to less than 1 micrometer. And we put one out here somewhere as a reference, and the second one is sitting here. And so we move it point by point toward the interface, and here's the distance scale. And if you're far enough away from the interface, the potential difference between here and here is 0. That's comforting. That's what you expect. But as you get closer and closer, you begin to pick up a negative electrical potential, which goes down to minus 120 millivolts. So the region of negativity seems to correspond roughly to the position of the exclusion zone. So it looks like the exclusion zone might be negatively charged. Now, we get rid of the gel and put a piece of Nafion there instead and repeat the experiment. The result is qualitatively similar, but quantitatively a bit different. The region of negativity extends farther out from the interface, but please remember that the exclusion zone is bigger in the case of Nafion. So there's a correspondence between the region of negativity and the exclusion zone. It looks as though the exclusion zone is negatively charged. But that doesn't really make sense if you think about it, because what you've done is you've taken some water and you've poured the water in, and the water starts out as neutral. So how do you start with neutral water and wind up with a fairly large negatively charged zone? Well, the only reasonable way you can do that is if you have a positively charged zone somewhere else. And the question is, well, is there any evidence for a positively charged zone somewhere else? And the answer is yes. If you look at the previous slide that I showed you and realize it's turned 90 degrees for convenience, and realize that you do have a negative zone, but remember the red color. The red color indicates a huge number of protons. So it looks like you have negative here and positive here. So the two seem to be in balance. And just to check to see whether there really is charge separation, we put one electrode here, and connected them through a resistor. So if there's charge separation, you should get current flow. And indeed, that's what we found. So the current starts at a fairly high value and goes down not to 0, but to some plateau value, which extends over some time. So it really looks as though there is definitely charge separation between the exclusion zone and the region beyond. So really what we have, what looks like a charged battery in water. And of course you don't expect a charged battery in water, but it appears that's from the evidence that's what we found. Now, one expectation of this, which we checked, is that if you have separated positive and negative charge, you should have a force between the two, because the two want to attract each other. And we actually tested recently to check to see whether there is this kind of attraction. And we did it by setting up a lever, which you see up at the top, a ribbon-shaped lever, and we put a piece of Nafion attached to it. And if you look at the second panel, you see what we expect to happen is that we have a negative exclusion zone here, and you have the positive charges that are out here. There should be a force between the positive charges and the negative charges that are attached to the Nafion. The exclusion zone is practically bound to the Nafion. So we expect the beam to deflect, and indeed that's what we saw. As you put the water in, the exclusion zone builds, the beam deflects. So it looks as though there really is our charges genuinely separated between the exclusion zone and the region beyond. So I've just shown you the red one, that the exclusion zone has negative charge and positive beyond. So now if you want to figure out what's going on in this exclusion zone, all of these are clues. But the three clues I think that are the most important, starting from the bottom, the blue ones, is that the exclusion zone molecules are aligned, and they're fairly stable, and they're constrained. If you put those together, what fits most closely is some kind of liquid crystal. So it looks as though this region is a liquid crystalline region, as opposed to the bulk water, which is a liquid. So far, what I've shown you is that you have a liquid crystalline region of some sort. You have negative charge in this region. This region excludes solutes profoundly, unlike bulk water. And there's some hint that it's non-dipolar because of reasons that I'll go into in a moment. Not just a series, a stack of dipoles. And it may extend very far. But very far, we're talking not the 2 or 3 molecular layers that the chemistry books will tell you, but if this is right, it's 2 or 3 million molecular layers that we see. Now, it was suggested 101 years ago by Sir William Hardy that there might be a fourth phase of water. So we think of solid, liquid, and vapor, but there were many reasons long ago forgotten that led to the suggestion that there might actually be a fourth phase. So, whether this is a fourth phase is up to you to decide or judge, but all of the physical and chemical properties of this zone are different from either solid or liquid. Now, what about the non-dipolar structure? Well, my hero, Gilbert Ling, the second person I showed you, suggested a stack of dipoles. But as we began to look at this, we realized that model is not adequate. The reason it's not adequate is that, first of all, is that this zone has negative charge. It has a lot of negative charge. Dipoles are neutral, so it doesn't matter how many dipoles you build up, stack, you still get neutrality. So that doesn't work. The second reason is that this 270 nanometer absorption of light in the exclusion zone, usually that kind of absorption is not associated with dipoles. It's associated with ring-like structures. So that's a second reason to doubt the stacked dipole. Well, if you want to figure out what the structure might be, where do you start? Well, the way we started is by saying, probably the structure of this zone should have some precedent. It should be built on some structure that we already know about. So what kind of water structure do we already know about? And that's ice. Okay, so this is a picture of a model of ice. And on the left side, these are just two different views of the same structure. The red dots are oxygens. And in between the two oxygens would be a hydrogen. It's just omitted to reduce the clutter. Now, if you look on the right side, you see these blue dots between the red oxygens, and these blue dots are protons, positively charged protons. And so what you have is that the negatively charged oxygens are, you might say, glued together by these positive protons, and that's why ice is hard. So we thought maybe some variant on the ice structure might be adequate to characterize the exclusion zone. And so the first idea is, well, let's just remove the protons. And if you remove the protons from a neutral structure, which is ice, take away the negatives, then you get, take away the positives, and then you get a net negative charge. And that's good because that's what we want. And also it's a non-solid. We don't want a solid. We want something that maybe has viscous gel-like properties. So it looked like a great idea until we realized, scratching our head, that it won't work. And the reason it won't work is that if you take away the positive charges connecting two negatives, then you have two negatives juxtaposed next to each other, and the structure would just fly apart. So a great idea, but unfortunately, it didn't work. So then, after a bit, we thought, here's another idea, very similar, but slightly different, which worked nicely. Take the two planes, shift one relative to the other by half of the oxygen to oxygen distance, then you get something that's very nice, because what you see is that you have the oxygen in the back plane, negative, juxtaposed next to the blue hydrogen, which is positive, so you have electrostatic sticking. So these planes then stick together nicely, but not as rigidly as in ice. So with this structure, you have a situation like this, where you have a material, it's sitting in water, and from the water molecules somehow build these easy layers that stack on one another one at a time, growing like this. And so that would be one way to account for this kind of structure. And if you look at one plane of this structure, it looks something like this. It's a bit like ice, but it's not exactly ice. However, if you count the number of oxygens and hydrogens, it's not H2O. It's actually H3, it's H3O2. So you might say that this zone of water, it's water, but it doesn't have the same chemical formula as water. It's slightly different, like H2O2, for example, hydrogen peroxide, same molecules, but in different ratio. And it's that ratio that confers the negative charge on this zone. Now, in constructing it, I took the top plane and moved it to the right relative to the bottom one, but there was nothing sacred about moving it to the right. I could have moved it to the left, or I could have moved it 60 degrees or 120 because of symmetry. and I would have gotten pretty much the same result. So the ability to shift in each of 6 directions gives a lot of latitude. For example, you can build structures like this, helical structures. And so here you've got layer 0, the first layer shifted this way by 0 degrees, 60 degrees, 120 degrees, giving you a helix. So why is a helix important? Well, helix is important because, as you know, in biology, most of the molecules are the fibrous proteins, for example, in DNA and RNA, they're all helical. And it's known that these entities are surrounded by some kind of ordered water. So it's possible that this creates a nice interface with those molecules. So the advantages of this, these sheet-like layers that stack is that, first of all, it's not pulled from a hat. It has precedent. It has negative charge, which is what we need. The ring-like structures are more able, with delocalized electrons, to explain the 270 nanometer absorption that's observed. It's able to accommodate helical structures. And an important one for chemists, that the large sheets eliminate the Brownian instability. Now, what do I mean by that? Well, if you look at the structure on the left, which you may think of as stacks of dipoles, you know, each dipole undergoes some kind of thermal motion, and if you start stacking them, the thermal motion builds, and you can't build more than 6 or 7 or 8 or 10. Various chemists have said, therefore, this kind of structure is impossible. We're not suggesting, there are arguments both ways. We're not suggesting this structure, we're suggesting a structure that has practically infinite extent, something like this. And when you increase the mass, the Brownian excursions diminish based on the mass. So we have something here that undergoes very small Brownian motions, and each layer is effectively glued to the region next to it. So the issue that you can't build many layers because thermal motion will make it impossible vanishes. Now, so that works. Now, another one is that the tight lattice keeps the hydronium ions out. So what do I mean by that? Well, we've got charge separation here. We have an exclusion zone that has a lot of negative charge, and we have a region outside that has positive charge. And as you know, when you have negative and positive, they want to do that. That doesn't happen, or it doesn't happen very much, because we can put the electrodes in, and you can put the electrodes at time equals 5 minutes or 1 hour or 3 hours, and it's pretty much the same. So how is it possible that those positive charges, which are actually not protons, they're protons latched onto water molecules to give hydronium ions, which we all learned about in freshman chemistry. Those hydronium ions, they really want to get desperately to move into here, but they can't move in. They don't move in, that's the observation, and so why don't they move in? Well, they don't move in, according to this model, because this lattice structure is so tight. The hexagons themselves are small, but the hexagons are shifted relative to one another, so the real openings are extremely small, so they can't get in. They keep separated. So the battery charge remains separated. So another advantage, this tight lattice keeps the hydronium ions out. So it looks like there are a whole bunch of reasons to think that this model is reasonable, but you might ask, well, okay, it's reasonable, but give me some evidence. Does it really exist? There are actually quite a few papers demonstrating hexagonal types of water near interfaces, but most of them deal with only a few layers because of the methods that have been used. But there's one method that goes more deeply, and that's shown here. The paper published by a Harvard group in 2008, and they studied the protein ATP synthase, the subunit C of that protein. It's a very old protein. It has an interesting character to it. What happens is that when the atmosphere is dry, it forms vesicles that you see, for example, in the top slide, that enclose the water. So you have a volume of water enclosed in this either circular vesicle that you see at the top, or different geometric shapes that you see here, but these are volumetric structures. So what these investigators did was to take an electron beam and do diffraction on the water. This is a volume of water in between. And the result was interesting. It's a series of dots, and the dots mean that the water is very well structured. And the fact that you have 6 dots around here means it's a hexagonal structure. So they concluded that the water inside these capsules are ordered hexagonal sheets of water. So the answer to question 2 is the easy physically distinct from the bulk. Yes, it's distinct, and it looks like it's a layered honeycomb structure. Okay, so now I presented to you this battery that we have, and as you know, batteries usually need charging or recharging. And the question is, what charges this battery? We couldn't figure this out for a few years. We scratched our heads, and finally we found out that, in fact, it was radiant energy, light. So light charges this battery. And we found out, actually, from an undergraduate student who was walking around. We had the chamber sitting here, and the undergraduate student had this light and was shining the light, and we saw that where he shined the light, the exclusion zone was building, actually rather impressively, something like you see here. Here are the microspheres, here's the exclusion zone, and this is the region that was illuminated. And you take the light away, and it comes back again. We found also, I won't show you the data, it's published, spectroscopic information. We found that the most powerful wavelength, among the wavelengths we studied from UV to infrared, about 5 micrometers, was the infrared, especially at 3 micrometers. And we could shine the infrared with a small light-emitting diode that emitted the infrared, and we could get an expansion easily of 10 times. You take it away, and it comes back again to its original. Now, where do you get infrared? Well, we're all familiar where you turn the range on, and it gets hot, and it emits infrared. But in fact, everything around us emits infrared. It's you might say, a gift from the sun or the stars, the cosmos, hitting the building and re-radiating on the walls and such. So this is essentially, this is free energy, literally free. It just comes to us. And this is the energy that goes into the water and builds the exclusion zone. So effectively, you have a situation like this. You have a material and water next to it. And this easy water, from the energy that comes in, many wavelengths, but infrared is the most important. It builds up like this, and if you were to take away the excess energy, it would come back. We also found a way of diminishing the ambient infrared energy. This took us, embarrassingly, a while to figure out, but if you take a simple Starbucks a coffee container, it insulates, right? And so what does that mean, to insulate? It means it blocks the infrared from coming into your iced coffee and warming it up. And so we took a dewar, the same sort of thing, but a professional one. We stuck the chamber with the exclusion zone inside. So this is the result. The top one is the control. You can see a standard size exclusion zone. And then we put it into the dewar for 15 minutes, took it out, and quickly looked before it built up again. And you can see that the exclusion zones on either side are smaller, roughly half the size, and then pull it out and let it sit on the bench, and they build up again back to the normal size. So it means that we can either increase the energy or decrease the energy, and it has a direct effect on the exclusion zone. So the answer to question 4 about energy is that EZ is powered by photonic energy, which orders the water and charges the water battery. Now, what about energy flow on the Earth or the solar system? So the sun hits the water and generates heat. We all know that. However, I've shown that it imparts energy for building order and separating charge. Now, if that's true, then you might ask, okay, if it's building up this potential energy, can you get the energy out after you've put the energy in? And we found that you can. We found that, again, a couple of undergraduate students found if you put a tube made of some hydrophilic material, either a Nafion to start with or a gel, that you get constant flow through the tube. And the experiment is shown here. You take the tube and you quickly you fill it with water to make sure there are no air bubbles. Put it in a water bath that also contains microspheres, so you can see if there's any movement. Put it in the microscope, and the result is shown here. So you see, so you have the flow, and the flow keeps going. And it's not just the Nafion tube. It's also, we found the same thing occurring in a gel. We cut a tunnel inside a gel, a cylindrical tunnel, and you can see it here that there's an exclusion zone, and right in the center are all the microspheres, And the video shows what happens. You've got constant flow through there. And recently, we tried to see if we add light, do we get additional flow? We found that we can actually increase the flow by up to 5 times by adding light. So this is flow that's basically free for the taking, and it's powered by light. So essentially, it means that this is doing work by driving it through because the water has some viscosity. So if work is done, if the conservation of energy works, then the system must be absorbing energy, and I suggested to you what the energy is. It's the electromagnetic energy that is coming in to give the potential energy to drive the work. So water transduces light energy. Okay. Now, again, if you're not Rupert Sheldrake, you'd think this is weird. However, however, if you think about it, think about the plant that's on your windowsill. It's doing the same thing. It's taking light energy and transducing that light into chemical energy, which drives metabolism, bending, growth, what have you. And so I'm suggesting to you that the same thing is happening in this glass of water right here. The energy is coming in, and it's getting transduced into some kind of potential energy from which you can get a lot of interesting things, some of which I don't have time to talk about. So that leads to the equation, and I know the units don't match, but that you can't really think about water without thinking of energy. Somehow there's energy that's getting into this, and this energy, again, I don't have time to suggest to you, but some of this energy may contain information. That's another story beyond this, but.